Hello, I'm Kristine Werling. I'm a PhD student at the Harvard T.H. Chan School of Public Health, and I'm in Flaminia Kataricia's lab. This is my first oral presentation, so don't be too hard on me. Today I'll be talking to you about my work looking at ecdysone receptor and how it regulates post-mating biology in Anopheles gambiae mosquitoes. So 20-hydroxyecdysone, or 20E, is a steroid hormone that's important for Anopheles gambiae reproduction. So a female requires a blood meal in order to produce her eggs, and in response to this blood feed, she produces a peak of 20E, and this 20E signal is essential for her to begin vitellogenesis, or genesis, so she's developing these eggs in her ovaries. And so each time she takes the blood meal, she produces this peak of 20E. And something else that's unique about some Anopheles mosquitoes is that they require 20E during a mating event as well. And so males will transfer 20E to females. And so during this mating event, a male will transfer sperm as well as a coagulation of seminal secretions in the form of a mating plug. And it's in this mating plug that 20E is transferred from males to females during mating. And previous work in our lab has shown that this 20E signal is both necessary and sufficient for inducing post-mating phenotypes in the female, in particular oviposition of her developed eggs, as well as a refractoriness to remating. And ecdysone receptor, or ECR, is a nuclear receptor that's the receptor for 20E that's been well described in Drosophila species as well as Aedes, particularly from work in the Reichel lab. And so this ECR receptor will form a heterodimer with ultraspirical, or USP, and together these have a high affinity for 20E. And so 20E can diffuse directly into the cell and will bind to this ECR USP complex, and together these will bind directly to DNA, and by recruiting co-activators or co-repressors can either turn on or turn off gene expression. And so we know a lot from Aedes that this ECR receptor is the critical receptor for 20E that's produced by the female after a blood feed. And so these two will bind together, and this interaction is essential for the process of oogenesis to begin. And so what I've done is looked at how, if ECR is also this receptor for 20E after blood feeding in Anopheles gambiae mosquitoes. And so we see that, in fact, this relationship is conserved. And so by knocking down ECR in adult female mosquitoes and then providing them with a blood meal, we can see more than a 50% decrease in the number of eggs laid in the ECR knockdown females compared to the controls. And if we also look at the level of expression of vitellogenin, which is an essential yolk protein precursor gene that's directly responsive to 20E and is highly upregulated following a blood meal, we can see in red that when ECR is knocked down and they're given a blood meal, this expression of vitellogenin is both delayed and reduced. So this will show that ECR seems to also be the critical receptor for 20E produced by Anopheles gambiae mosquitoes after a blood meal. But since Anopheles gambiae mosquitoes also require 20E during a mating event, we wanted to know whether ECR is also this receptor that's important for mediating this 20E signal that's transferred from males to females during mating, and whether this is essential for induction of post-mating phenotypes, oviposition, and refractoriness to remating. Another question that we have is that since these mosquito species have two 20E signals that are coming both after a blood meal and during a mating event, how does the female keep these two pathways distinct? So if she's taking a blood meal, how does she keep that only for egg development and it doesn't prematurely activate post-mating phenotypes? And so to first address this initial question of whether ECR is the critical receptor for 20E produced after a blood meal, transferred during mating, I again did knockdowns of the ECR receptor in 1-day-old adult female mosquitoes, and then mated them with wild-type males. And so by looking at both the transfer of a mating plug in the atrium as well as presence of sperm in the theca, we can see that, as we expect, ECR doesn't have any role in mediating their ability to mate a first time. But the more interesting question is whether it affects their refractoriness to remating. So by taking these females who have already successfully mated once and then mating them again to a second group of males, we looked at their ability to be mated a second time. So by looking again at their ability to transfer, to receive a mating plug. Now we see what we expect in the control group, which is that nearly 90% of the females are unable to receive a second mating plug, which means that they've become refractory to remating after their first mating event. But we also see that by comparing the control to the ECR knockdown, there's no difference between the two. And so this does suggest that ECR is not the critical receptor for mediating refractoriness to remating in Anopheles mosquitoes. But then also looking at the second post-mating phenotype, which is oviposition, following the same experimental setup of knocking down ECR and then mating them, I looked at their ability to oviposit their eggs in the absence of ECR. And we see the same story, that there's no difference between the control and the ECR knockdown females. So for those females that developed their eggs, 100% of them are ovipositing in both the control and the ECR knockdown females, which is a suggestion, again, that ECR does not regulate oviposition. The only time that we do see an effect of the ECR knockdown is when we look at the timing of oviposition. So here we have days post-blood feed and how many females have oviposited their eggs. And so we see a slight one-day delay of oviposition in the ECR knockdown females. But we think that rather this being an inhibition of oviposition, it's probably due to their delayed vitellogenesis and likely also delayed egg development. So taken together, by looking at both these refractoriness to remating data and oviposition, it seems to be that this ECR receptor is not critical for regulating post-mating phenotypes. So it may not be the important receptor in the mating event. And so briefly, what I've shown you so far is that we see a conserved interaction between ECR and 20E following a blood meal in Anopheles mosquitoes. But when we look at the role of ECR in regulating 20E signaling after mating, it seems that this ecdysone receptor is not the critical receptor for 20E signaling following a mating event, as it doesn't have any effect on their ability to oviposit or become refractory to further mating. And so this leaves us with the question of what is this receptor? So who is responding to the 20E signal that's transferred during the mating event? And so this is certainly something that we're going to be looking in going forward for what this other receptor might be. Now moving on to the second question that I put forth is why doesn't a female 20E induce post-mating responses? And so as I described in the beginning, we have two 20E signalings that are happening during an adult female's life. And you can imagine that if a virgin female takes a blood meal, she's producing this peak of 20E. And if this 20E signal were to prematurely activate these post-mating phenotypes, then you'll have a virgin that's now become refractory to remating. And certainly that she doesn't want that to happen if she wants to be reproduced and create many offspring. So why is it that this ECR-20E interaction prevents these from happening? So is this ECR receptor a critical component of keeping these two pathways distinct? So this is one question that we had. And so by, again, knocking down ECR in adult female mosquitoes, I now provided them with a blood meal. And so here they're producing 20E in the absence of the ecdysone receptor. And in fact, we see that when ECR is depleted, these females are now induced to oviposit after having just taken a blood meal. So now we have virgins that are ovipositing sterile eggs, but showing this post-mating phenotype. So here we have between 40 and 50% of females that are ovipositing. Now when we reverse the order of events, so if we're providing the females first with a blood meal and then knocking down the ecdysone receptor, we don't see this phenotype. And so this would again suggest that when this 20E signal from the blood feed is happening in the presence of the ecdysone receptor, you have protection of these post-mating phenotypes, so you don't see induction of oviposition. But again, when the signal is coming without the ecdysone receptor, you see these pathways activated. So again, in addition to looking at oviposition, I also wanted to look at whether this ECR-20E interaction was also interfering with their refractoriness to remating phenotype. And so by knocking down ECR and again providing a blood meal, I looked at their ability to mate. And so if this 20E signal is able to activate post-mating phenotypes in the absence of ecdysone receptor, we might expect that these females have now become refractory to remating, even though they are in fact virgin. And so here, looking at just the presence of plug, so their ability to receive the mating plug, we don't see any difference in their ability to receive the mating plug between control and knockdown females. But when we take a closer look at what's happening, it seems that plug transfer actually isn't normal in these ECR knockdown females. And so here we have the female atria of the control females, and they're receiving a single mating plug, and it's oriented such that the tip of the mating plug is directed towards the opening of the theca duct. But when we look at the ecdysone receptor females, we see a different story. We often see that they're receiving multiple mating plugs, and these plugs are jammed up close to the ampulla, and they're not pointing in the direction of the theca. So they're very different from our control females. And so one thing that we know about females after they mate is that they undergo atrial remodeling, and we think that this might be the process that's essential for them to become physically refractory to further mating. So what we might be seeing here is that these ecdysone knockdown females have undergone some atrial remodeling such that they're no longer able to receive a mating plug in the way that they typically would. And then when we look also at sperm storage of these females, we're looking directly into the theca to see if they have any sperm in their theca. We see between 60 and 70% of these females don't have any sperm in their theca. And so we do see the sperm being transferred, so there's accumulation of sperm in the atria, but immediately after mating and several days after mating, that sperm never makes its way into the theca. And so by looking at both this abnormal plug transfer and the absence of sperm in the theca after a mating event, it would seem that in the absence of ECR, this blood feeding is also inducing refractoriness to mating, particularly at the level of insemination. And so by considering both the induction of oviposition and the refractoriness to remating data, it would seem that the ECR receptor has a critical role in post-mating 20E signaling, and that it's essential for keeping these two pathways distinct from one another. And so briefly, just to review what I've talked about today so far, is that ecdysone receptor seems to regulate this 20E signal produced by female after a blood feed, which is something that we would expect looking at other mosquito species. But it seems that ECR does not have a critical role in regulating 20E signaling after a mating event, and so this leaves the question of what this alternative receptor might be. And finally, that the interaction between ECR and 20E produced after a blood meal is a critical interaction that prevents this 20E signal from a blood feed from activating post-mating phenotypes in virgin females. And so we're certainly investigating what this mechanism might be, but one idea is that the physical interaction between 20E and ECR prevents this 20E steroid hormone from possibly getting to other tissues and activating alternative receptors. Another possibility is that this ECR ecdysone complex together actively repress post-mating phenotypes following a blood meal. But certainly we're still doing work looking towards the alternative receptor and what the mechanism might be here. And so those are ongoing questions that we're working on. And so with that, I'd like to thank the Catarruccia Lab, in particular Evie, Sarah, Maurice, Matt, Paolo, and my mentor Flaminia, as well as my PhD program at Harvard T.H. Chan, funding sources, as well as ASTMH Travel Award for supporting my trip here today. Thank you. I'll take any questions.
All right, we have time for questions. Okay,